I'm Hongbin Liu from the Shenzhen Institute of Advanced Technology, Chinese Academy of Science. Today, I will present our work on exploring human gut microbiome functions based on the structure proteome. Many hosts, including animals and plants, have symbiotic microbiomes that influence host health and disease. Understanding how these microbiomes impact host physiology has been a long-standing question, and science has highlighted this as one of the most pressing questions in global biomedical research. Despite the substantial research process in human gut microbiomes, significant knowledge gaps remain, such as uncharacterized microbial species and genes with unknown functions. It's estimated that about 40% of microbial genes lack functional annotation. Our analysis of the human gut microbiome corroborates this estimate. Using sequence-based approaches, we found that about 80% of phage proteins and 30% of bacterial proteins still lack functional annotation. Notably, protein structures are often more evolutionary conserved than sequences. This suggests that proteins with similar functions may adopt similar structures even when their sequences differ. Therefore, structural similarity can serve as a proxy for functional similarity. Based on this principle, we proposed exploring human gut microbiome functions through a structure proteome approach. We constructed a comprehensive human gut microbial protein structure database, GMPS, covering 2.7 million predicted protein structures from phage genomes and bacterial genomes. We first focused on phage proteins. We expected to improve annotation by comparing and classifying GMPS phage protein structures with no protein structure databases based on structure similarity. Using a combined sequence and structure classing strategy, we generated structure clusters in which proteins share significant structure similarity. Within each cluster, proteins with annotated function can help infer the functions of structurally similar but unannotated phage proteins. This approach increases the functional annotation coverage of gut phage proteins from 25% to 50%. This method not only improves function inference for individual phage genome, but also allows systematic analysis of protein families of interest. For example, we focus on phage encoded enzymes with bacterial lytic activity, phage lysins. During the late stage of phage replication, assembled phage secrete lysis that degrade bacterial peptidoglycan, inducing cell lysis and releasing offspring phages. Phage lysis typically contain two domains. A catalytic domain that EAD responsible for the activity, and a cell binding domain CBD responsible for the recognition. Through structure classing, we found that the catalytic domain is the co-functional domain of lysis, and the combination of EAD and CBD domains shows significant domain shuffling. And for example, lysin from Clostridium inoculum phage combines EAD type found in streptococcal phage with a CBD type found in Bacillus phage, illustrating how domain shuffling diversifies lysin structures. With the selected phage lysins targeting gut pathogens for functional validation, for example, Clostridium inoculum and Enterococcus gallinarum. These lysins demonstrated strong in vitro activities, suggesting their potential as novel antibacterial proteins. Notably, these lysins share less than 10% sequence identity with no lysins, underscoring the limitations of sequence-based discovery methods. We also observed the diversity in license specificity. All of these licenses showed high specificity against target specific species, but show variable effects on non-target gut species. High specificity license enable the target the precise removal of target species with this slide has some problems. Although study of species specificity, ecology, and metabolism, yeah, proof of concept experiment, we constructed a synthetic gut community and added a license. After several passages, target species were accurately removed, confirming license potential for precise microbiome modulation. The next, we focused on bacterial proteins using structure comparison and structure-aware AI models. We explored new bacterial proteins. We focused on microbial-host enzymes widespread in gut microbes that modulate host physiology, but often lack sequence conservation. And systematic analysis confirms that cross-domain isozymes generally exhibit show low sequence similarity but remain structure conservation. For instance, when searching for glutamate decarboxylase homologs in bacteroides, sequence-based ranking placed the right candidate over 700, while the structure-based ranking identified it as top-ranked, highlighting the power of structure for remote homolog detection. We applied this approach to microbial melatonin synthesis, which involved three enzymes, DDC, ANT, and ASMT. We first identified activity for all three enzymes through culture of fecal community, and they will grade these enzyme activities in isolated human gut bacterial strains and confirmed their activities in gut microbials. Bifidobacterium adolescens show DDC and ANT activity, while Bacteroides story showed ASMT activity. Using human enzymes as structure templates, we identified microbial homologs through structure comparison, followed by experimental validation. These enzymes showed high structure similarity, but low sequence identity to human templates, demonstrating the advantage of structure-based detection. Heterologous expression and gene knockout and mice experiments first confirmed that gut microbial enzymes contribute to host melatonin levels, potentially influence host mood and sleep. However, not all functionally similar enzymes shared a global structure similarity. Our dataset revealed that a substantial fraction of microbial host enzymes are globally structured dissimilar, making traditional global structure comparison insufficient. To address this, we developed DDER, a structure-aware AI model for remote enzyme retrieval. DER combines protein allopoid models with constructive learning and embedding-based retrieval to identify remote isozymes. On both the precision and recall metrics, the outperforms existing sequence and structure-based methods such as MMCs, +P, and USLI and FOSIC, while providing substantial speed advantages. In summary, we provide two resources for remote homolog detection: the GMPS database, the human gut microbial protein structure databases, and structure-aware AI models for remote enzyme discovery. Using this, we discovered the novel phage license with bacteriocity activity and microbial melatonin synthesis enzymes. Finally, I would like to thank the lab PI, Professor Lei Dai, as well collaborators Kai Wang and Chang Taojiang from Peking University, and Yu Li from the Chinese University Hong Kong. This is my email, and any comments and questions are welcome. Thank you for your listening.